Eccoci qua, partiamo. Buongiorno a tutti e grazie per la partecipazione. Per chi non mi conosce, io sono Linda Croci e sono una dei responsabili commerciali di RT Environment, oggi nella veste di moderatore. Prima di iniziare rubo giusto un paio di minuti per una breve presentazione aziendale. RT Environment, marchio Watec, è un'azienda italiana con sede a Veggiano, Padova, specializzata nella distribuzione e manutenzione di strumentazione e sistemi per il monitoraggio delle acque. Operiamo come distributori in tutta Italia di vari marchi, tra cui di YSI appunto Aanderaa, entrambi del gruppo Xylem. E oltre alla distribuzione offriamo anche servizi di noleggio, manutenzione, assistenza tecnica e supporto all'installazione. Siamo presenti in tutta Italia con responsabili commerciali distribuiti per regione. Sicuramente conoscete i miei colleghi Gianluca Presutti e Francesca Accardo. Vi invito a visitare il nostro sito internet che stiamo aggiornando ogni giorno, www.watec.it. Adesso passo la parola ad Anders Stenberg, molti di voi già lo conoscono, è connesso direttamente dal campo in un magazzino in un porto e un'ultima cosa, le domande le potete porre direttamente nella chat. Hi Anders, welcome, now you can start.
Can you see my screen?
Yes.
Ok. Buongiorno a tutti. Mille grazie for this opportunity to present, and thank you for organizing. So yeah, I'm sorry about not being dressed up. As Linda said, I'm just had to urgently go to a harbor and try to fix a buoy, which is part of the navigational system, Swedish navigational system. But the presentation will be about how to study processes at the seafloor and different methods, or how you use one method that we have applied for over 20 years now, and that is using bottom landers, benthic landers, to study the importance of the seafloor in different situations. This presentation will largely contain information from which is presented in a paper which was actually released in January this year, and it's a summary of our experiences using bottom landers in different types of projects. And here I want to recognize, of course, all my co-authors here, or the main author is Mikhail Kononets. He is now working with our landers at the Gothenburg University. And then also I want to point out Per Hall, who is the leader of our research group. We focus a lot on processes, but we also have other projects running. So in this paper, there is a listing the scientific publications that we have obtained with using our bottom landers, and it's about 30-35 publications, and you can see a list of them here. After the presentation, Linda will send you this paper and also the presentation as a PDF. In the presentation, I will focus on some things: oxygen respiration, on benthic nutrient cycles, speak a little bit more about phosphate, which we have been working quite much on, because that's a major issue in our region, in the Baltic Sea, for example. How to use modeling results combined with the benthic flux measurements from the lander and with long-term monitoring. I will show you one or two projects about that, and then talk a little bit about the technology that we use on our landers. And the landers, we have 3 landers, and here is what we call the big lander. We have done more than 300 deployments in total Now I think it's more like 350, more than 1,000 incubations. And we have deployed from water depths ranging from 5 to 5,600 meters. And what the landers basically do when we use them in the incubation mode, because we can also put other modules on them, then they take water samples that we analyze for concentration changes over time and calculate fluxes from. And we have sensors, 15 to 25, all comes from Aanderaa, the company that I work for. I also work for University of Gothenburg. And through the years, we have been using them for about 20 years now. We have about 95% success rate in the deployment. Sometimes there is a chamber leaking, sometimes 1 or 2 syringes does not trigger as they should. And as I said, about 35 scientific papers have been used using these tools. And what you see here is the main lander with the carrier frame and ready to be deployed with the railway track, so ballast weights, which makes it sink to the seafloor, and here with floats, which makes it easy to recover once it is at the surface again. So basically it sinks to the seafloor. The lander stays there for a couple of hours. Then the incubation chambers are gently pushed into the bottom and lids are closed. And then you start your measurements. And the chambers we have, and then after normally depending on how active the sediments are, normal deployments for us is between 10 and 48 hours. 48 hours on very low activity sediments like we see here in the deep Arctic or in the Mediterranean Sea. And it depends on where in the Mediterranean Sea, of course. And shorter incubation times when we work in more active sediments with more organic rich sediments. So after deployment is finished, the lander takes sediment, recovers the sediment, and then you send an acoustic signal to it. It releases the weights and it rises to the surface where you can then recover it. And then it has different systems for positioning underwater and also for positioning when it is at the surface. So and this picture is actually from the deepest deployment that was in the Arctic. The deepest place in the Arctic, it's the Molloy depth, 5,600 meters. And here is an example then of the incubation chambers. The big lander carries 4 incubation chambers like this. So once at the seafloor, the chambers are gently pushed into the bottom. A lid is closed. Stirrer starts, and the stirrer has is doing 2 tasks. One is to avoid that you have gradients forming inside the incubation chamber, and the other is to try to mimic hydrodynamics inside the chamber, mimic what you have outside, which is basically impossible, but you can do some kind of rough estimates of how strong currents are outside and then try to mimic that with inside the incubation chamber. And here you see each chamber has 10 syringes. They can either be used for injection. We can inject tracers and do experiments. Algae, for example, look at the breakdown of the algae, different things. And they can also sample water samples. And then we have sensors inside the chambers, and I will get back to that, what we do with these. So basically what we do, we measure exchange between the sediment and the overlying water, what we call benthic fluxes. And here are parameters that we have studied through the years, oxygen and total carbonate. For total carbonate, oxygen we measure with optical todes. Total carbonate, we take samples and analyze in the lab. And then nutrients, and these we analyze in the water samples. We don't have any way to measure them in situ. pH, we have used pH sensors sometimes. Methane, we take samples also for alkalinity, dissolved organic carbon, and also for these parameters here like iron and manganese. And these are arsenic, and these things here are more for contaminated sediments, which we have been involved in some projects, and I will come back to that. Sensors we have also inside the chambers is salinity sensor, and I will come back to why. And PCO2 we have used, but we're not using anymore. We have some issues with the calibrations of these sensors. And turbidity we have to create resuspension experiments by stirring more rapidly. So in the previous picture, you saw how the lander is used when it's autonomous, when you deploy it and it sinks by itself to the seafloor, and it comes back again when you release the ballast weights with an acoustic release. Here is pictures of how these landers look when we just use them in shallow water, so down to about 150 meters. Then we lower the whole incubation frame here. This is with 4-module frame, and this is a 2-module frame. We lower it on a rope gently, and then the weight of the frame in the water is about 60 kilos, something like that. And with the incubation chambers open, and then data logged on one single logger. Here is a Seaguard logger, which logs all the sensors. And here is another image of how these chambers look. And you see the sensors that we use inside the incubation chambers as a routine. That is salinity, oxygen, and turbidity. And this one here we didn't use yet because this is a new turbidity sensor coming from Aanderaa. And but we use the older type of turbidity sensors. And we also started to use pressure sensors recently, and that gives us a very good way, as you will see, of detecting if syringes are triggering as they should, if stirring is working, when sediment is taken, when the lid is closed, and it gives feedback. So here you see an incubation chamber after recovery of the lander. You can see syringes have been triggered. I have taken water samples. This one was used for injection. And there is sediment in the incubation chamber, which has been recovered then from the bottom. So I show you now examples why we use, for example, salinity sensor inside the chambers. And you can see it here. This is in the Baltic Sea, salinity is very low. In the beginning of the deployment, we inject milk water into the chambers, and that will lower the salinity. You can see here black, red, green, blue. These are the four chambers. How salinity is lowered inside the chambers, and it stays low during the deployment. This line here, the more brownish one, it's the salinity outside the chambers. Why we inject? Because if you want to calculate fluxes, exchange rates between the sediment and the overlying water, you must know the volume accurately inside your incubation chambers. And by injecting a known volume of milk water and simple dilution calculations, we get how big the volume is, plus minus a couple of % inside the incubation chambers. In addition, we get a very good weighting if the chambers are leaking or not, because it can happen a stone below the chamber, it's not well inserted into the sediment, that we can see immediately if on the salinity, because then salinity starts to follow outside salinity, and also it rapidly goes up again. So we see injection of salinity here. This is a deployment where we first make one incubation with low stirring and look at the oxygen concentration. Here you can see oxygen concentration decreasing inside the chambers with low stirring and no resuspension of sediments. So again, the 4 chambers here in different colors, and brown is oxygen outside. And halfway through the incubation, we open the lid automatically and ventilate out the chambers. And then we start a new incubation. You can see oxygen decreasing. Then we don't do any injection of salinity. We just start a new incubation. And with much higher stirring, which creates resuspension inside the chambers. So this enables us to look at, to compare what kind of effects does resuspension have on release of nutrients, on oxygen consumption, on release of CO2, for example, or DIC from the seafloor, inorganic carbon. Without resuspension, as you see here on the left, and with resuspension here, as you see on the right. And we can see here, the plot here, that is the oxygen flux calculated for every value. Basically, it's dt. It's the oxygen flux calculated for every minute here during the incubation. Here you see fluxes of between 10 and 15 millimoles per square meter a day. But when we increase the stirring to really higher stirring and resuspension, mimicking, for example, natural resuspension or resuspension by some activity, then we get much higher flux rates, about the factor of about 5 millimoles per square meter a day, something like that, higher, 50% higher, and then it decreases over time. We can also mimic man-made resuspension from trawling or dredging when we take the sediment. By not dampening the sediment taking, we take up the sediment and shake it on the seafloor, and then we can mimic man-made or more violent resuspension. I told you we recently incorporated pressure sensors inside the chambers. This is a pressure sensor which is 6,000 meter rated. So you can see here why we do it. It gives us really good feedback on if everything that we programmed the lander to do with the processor is working as we expect. We can see here lid closed, that the pressure jumps up, and when one of the syringes injects, it jumps up. And then when the syringes sample, you can see that that also gives what looks like spikes, but when you zoom in, you can see that this actually lasts for 1 or 2 or 3 minutes, something like that. It's the time for the pressure to stabilize after injection or lid closing or syringe sampling. And at the end here, you can see when we take the sediment, the chamber goes up about 30 centimeters, and then here it rises, the lander rises to the surface. So it gives us really good feedback with the salinity and the pressure. We have really good quality control of how the lander is working, and we check that when the lander gets up. It takes about 5 minutes, and then we can say if a chamber is leaking, we don't take the water samples, we don't analyze the water samples. If everything looks fine, then we take the water samples and proceed with analyzing and with the experiments, or with the analyzing of the experiments. So how to log the sensors is with one logger on our landers. They are very old. They are about 20 years old. But on more recent landers, like this one from NIO, National Institute of Oceanography, in Goa, India, they use the Seaguard to log all sensors on their lander. This lander is commercially made by a company in Denmark, which is called KC Denmark. And in this case, one Seaguard instrument logs all sensors. In this case, free turbidity, free oxygen, free salinity. So in this case, there is 2 chambers. So one sensor in each chamber of oxygen, turbidity, and salinity, and one outside to measure the conditions outside. They also have chlorophyll, and they have a wave and tide sensor, and they measure currents. They can either put a single point current sensor on or a profiling current sensor to get the currents in the water column and higher up above the seafloor. And the sensors here, they can just be added, plug and play, as long as you have connections. And basically, the biggest systems we have delivered, they have about 50 sensors or so. And another advantage with this newer Seaguard 2 platform is that it can log sensors at different time intervals. So for example, pressure, you need to log at maybe 5-second intervals to pick up well the triggering of the syringes. And then current sensor, maybe you log at 10-minute interval to save power. and other sensors typically you log at 1 minute interval or so. So now to one of the projects where we have worked with our landers and where we have combined long-term measurements over 1 year at a time with moored systems close to the bottom, with expeditions using our landers and with modeling. So this was a project in the Gulf of Finland, which is a part of the Baltic Sea. It's one of the most eutrophicated parts of the Baltic Sea. We have St. Petersburg here, the biggest city in Northern Europe, and I think second biggest city in Europe, actually, after Moscow. Or maybe London is bigger, I'm not sure. But yeah, about 6 million people here. A lot of, and the biggest river coming into the Baltic here, it's the Neva River coming from the Ladoga Lake. So this Gulf of Finland is about 300 kilometers long, and these were the main stations we worked at. And the hypothesis when we started this was that there has been steady increase of nutrients in the water here over the years. And when perestroika came in 2000 or 1990, then the agriculture on this side here, which is Estonia, Soviet Union, part of Soviet Union, former Soviet Union, and Russia, agriculture collapsed, and there was much less input into the water from land. Still, nutrient levels were increasing here. So our hypothesis was that the nutrients were released from resuspension of sediments. So we deployed at 3 sites here at the mouth of the Gulf, in the middle of the Gulf of Finland, and more close to where the Russian border is. Instruments at about 60 meters depth, this type of instruments. So it's an earlier version of the Seaguard instruments we have. Was called RCM9. And they had, for the first time, optical oxygen sensors on, and measuring. They were deployed about 0.5 meter above bottom, and we had one turbidity sensor at this instrument and another one on a stick higher up. And we wanted to see at what currents you will have resuspension and what was the effects of sediment resuspension here. So that's why we have a gradient of turbidity, and that enables you to calculate how much of the bottom layer is resuspended. And then we did a lot of resuspension experiments with the landers in situ and in the lab on board by taking samples and running critical shear stress experiments and a lot of other experiments looking at how flux has changed with resuspension. So actually, after recovering instruments which were moored for 1 year at these 3 sites and looking at the data, we realized that resuspension had not much to do with the eutrophication here, at least, because currents were quite low and resuspension only happened a couple of times in the year. But what was really happening here was a really big variation in oxygen. You can see going from very low conditions in the late summer or August or so, to higher in the winter when winter storms and so kicks in and mixes, and then gradually decreasing during the spring until the summer when you have really good low conditions. So what you see here, the colors are oxygen measurements then at the different stations. So yeah, I said 70 meters, but it's actually around 50 meters here. Red is the station at the mouth of the Gulf, green is in the middle, and yellow is closer to Russia. And this level here, you have what is considered to be bad conditions for living animals. So you can see that over large periods of the year, we have really low oxygen levels, so low that only specialized animals can survive, animals that tolerate low oxygen hypoxia. And if you really zoom in to a period like this, you can see how variable it is. They have been taking water samples here monthly but these water samples is of very little value because depending on if you go out here or you go out here, you will get either excellent oxygen conditions or really bad oxygen conditions. So it's a matter of a week's time when you do your sampling. So what we could discover was that what was happening here is that when oxygen goes low, and that's known since before, when oxygen goes low, phosphate is released, and phosphate is the main driver here of eutrophication. With lower phosphate levels, we wouldn't have the blue-green algae, for example, in the summer. And you can see here how we can do experiments on that in situ using the lander, because we can then we deploy the lander and let the chambers go anoxic. And we can see then, so this is the oxygen levels here in red, and the time is about 20 hours in this case. You see how oxygen is decreasing over time till it goes anoxic. And blue is syringe samples, taking water samples that we analyze for nutrients, in this case phosphate. You can see when you go below about 10 to 20 micromoles of oxygen, then the phosphate release increases. And this is because the binding between iron and phosphate then goes away, and phosphate is released to the overlying water. And you can see also that other fluxes seems to decrease when oxygen is disappearing. We can see ammonia flux, for example, becomes lower, and also the silica flux becomes lower. And so we use long-term measurements to see how variable oxygen, salinity, temperature, currents, and so were, and to calibrate models. 3D model about eutrophication and circulation in the Gulf of Finland that was developed by University in St. Petersburg. And so we used the instruments to calibrate the model and validate the model. And then we used our flux measurements to estimate how much is the phosphate or nutrient release from different types of sediments at different oxygen conditions. So by doing this, we calculated the number of days that oxygen was below 20 micromoles per liter, because that was when you really had increase in the release of phosphorus from the sediments. And then how much of phosphorus was released from the different areas at the different conditions, oxic or anoxic water conditions. And the conclusion that we came up with from this modeling and calculations combined project was that the internal load, the phosphorus coming from the sediments, was about 66,000 tons per year, which is about 10 times more than the input from all the land-based sources, which then brings up the point that which also explains why phosphate was increasing in the water column, even if input from land was decreasing, because the major source of phosphate was from the sediments. And that's now generally accepted for the Baltic Sea, at least, that there is a big pool of phosphate in the bottom sediments, which is released when oxygen goes low. So as said, we can do experiments with the landers, and here is one experiment we did. We have also injected other things to study the nitrogen cycle, for example, to look at breakdown of blue-green algae at the seafloor. But here is another experiment where we injected marl. Marl is a byproduct from cement production, and there's a lot of that on an island in the Baltic Sea, which is called Gotland. And it's a lime-rich clay. And laboratory studies had shown that at anoxic conditions, this could bind phosphate. So what we did, we deployed landers on the bottom of at about 100 meters in one of the fjords in the Stockholm archipelago, and injected marl in some of the chambers. And here you can see the results from the syringe samples. So phosphate is leaking out from the sediments. The triangles here, they are controlled. There is no injection of marl. and the diamonds, they are injection of marl into the chambers, and you can see that decreases the release of phosphate from the sediments. More laboratory experiments are ongoing here, and to see if this could be a way to bind phosphate in some areas in the Baltic Sea. But of course, then you also must look at the risks of adding such a thing to the sediment. So also in the beginning, I talked about arsenic fluxes, and here is another example of where we combine water column monitoring with measurements of fluxes with the landers, and with creating decision support model for risk management of dumped chemical weapons, and also of oil leaking wrecks. This is a work that I have been part of inside with another university, which is University in Gothenburg as well, a technological university. And you can see here, after the Second World War, the Germans had big stocks of chemical weapons, mainly mustard gas, and the Allies wanted to get rid of it. So easy way was to throw it in the sea. So there was a lot of this, about 100,000 tons dumped in the Baltic Sea. Some the Russians dumped mainly like this, the barrels like this, and the British and the Americans, they filled bad ships with the grenades and the barrels, and towed them offshore and in deep water, and torpedoed them or sank them. And off the west coast of Sweden, there is one of these convoys had bad weather, so they sunk it beforehand. They were supposed to sink it in the deepest area in the North Sea, which is more closer to Norway. But it's lying there, and we wanted to see how resuspension of sediments and activities like trawling will affect the spreading of contaminants from the grenades if they were corroding, and they are corroding over time, and you could see on the pictures how corroded some of them were. And so first we looked at, we deployed instruments at the bottom. This area here, there's about 200 meters depth. We deployed acoustic profilers at the bottom with turbidity sensors and oxygen and salinity. And here you can see the response from a turbidity sensor on one of the instruments. You can see that there are peaks in turbidity over time here. This is just 2 weeks. And these peaks are all related to trawlers. So trawl here because there are wrecks, there is structures, then there is more fish. So you can see the peaks in turbidity and the instrument close to the bottom, because the instrument was placed about 0.5 meter or 1 meter above the bottom. Then by combining the turbidity measurements, so here you can see the trawling tracks. These brown dots here, these are where there are known wrecks and ammunition. And here you can see a wreck on a side scan sonar, and you can see also how the trawling tracks here from the side scan sonar. And to look at how high above the seafloor the sediments are resuspended, because currents here were too low to resuspend sediments. We can see here that by combining the acoustic signal strength, the backscatter from the acoustics, with the turbidity sensor, we get a kind of an internal calibration of the acoustics. And we can see that every peak from the trawlers, also you can see it in the backscatter. And that tells us that sediments are resuspended about 20 to 30 meters above the seafloor, and preferably it's the finer sediments like clay which are resuspended. And then we combine this with data that we got from the Swedish Geological Survey on arsenic. Arsenic was part of the mustard gas to make it possible to use in the winter, and also to make it more toxic. And the highest values that Swedish Geological Survey found in Swedish sediments was in this place here. That was the highest value in all Swedish waters that they found. The currents that we measured close to the bottom, about from the bottom to 25 meters above, was in this direction here. Currents are going north west, and the dump site, it was here where you see the flag, or it is here where you see the flag. So it's directly downstream of the dump site, and so basically we think that the trawlers resuspend sediments with mustard gas, some mustard gas, and it is transported with the currents with the fine material. And so we tried with the authorities to try to get a ban on trawling in dump site, this dump site, but that's very difficult politically because it's also outside the territorial waters. It's inside the Swedish economical zone, but not in. It's outside the territorial 12 nautical mile limit. And then Swedish authorities have been fishing here and looking at if there is traces of chemical weapons in, and they could find traces of. We got help from institute specialized in chemical weapons analyzing in Finland in one of the projects here. And they found the traces of Clark 1 and Clark 2, which are only found in the mustard gas. So it's they found small amounts of that in some of the fish and shellfish from the dump site here. So it's basically spreading to the environment. We can see very low amounts so far, but of course this is not a good signal. So we combined this with measuring with the lander, but the lander measurements were done in the Baltic Sea at another dump site or close to another dump site, measuring metals and including arsenic from the sediment. And what we did here was that you can see on all these parameters that we measured, molybdenum, uranium, which one is that now? You probably know, manganese, and then arsenic. We had always in this incubation a blank chamber to see that we were not contaminating our incubations. The blank chamber was basically a chamber covered with plastic, not in contact with the sediment. So you can see that they don't give much flow, or don't give any fluxes, while the others give fluxes. And here we measured quite high fluxes from the sediment of arsenic. So to summarize, we have used autonomous bottom landers for more than 20 years now, and we have continuously tried to improve them, to improve the quality of the measurements we do, improve the how easy they are to use and maintain, and to recycle on the deck. For example, now students, one student and one more senior can go out and do measurements and do a lot of incubations with them. We get high quality, consistent results by combining sensors inside the chambers with syringes and injection and sampling. We think it's in the projects we have worked, it's very useful to combine long-term bottom monitoring with lander incubations, which then lasts for shorter expeditions like 1 to 3 weeks, something like that. And from the multiple resuspension experiments we have done, we have seen that oxygen resuspension plays a role mainly on oxygen consumption, not so much for the release of nutrients from sediments. And as we have seen from the Baltic Sea example, and we have seen that several other places as well, that release of phosphorus is governed by oxygen variations, not so much by other things. I showed you some examples of trying to bind phosphorus at the sediment with marl. And we could measure fluxes of arsenic from sediments contaminated by chemical weapons. And with this, I would like to say grazie for listening. I hope it was of some use to you. And maybe Linda, you have, I don't know if there are any questions.
Thank you, Anders. Grazie mille. Ci sono domande? Le potete scrivere nella chat. Non ci sono domande.
Or can I not unmute and talk?
Ok, intanto che qualcuno scrive domande, nella speranza che qualcuno scriva domande, spiego un pochino di follow up, vi invieremo un'email con la presentazione, la registrazione. Colgo anche occasione per anticiparvi che questo qui è in realtà il primo di 5 webinar che svolgeremo nei prossimi mesi. Il prossimo sarà a marzo, l'argomento è ancora in fase di definizione. Non vedo domande. I can't see any questions, so I.
And if someone is interested in some of the publications, some more information, except for that paper that you will send out, Linda.
Yep.
Yes, you can just contact Linda or contact me.
Oh, a question. Anders, can you read the question?
Yes. Let me see your chat. Now my chat disappeared. Where is it?
Okay. How do you solve the problem of the air in the syringe?
So what we have so we can see. Can you still see the PowerPoint? Yes.
Yes, the one. Yes.
Yeah. So if we look at the picture of this, maybe you can see it here. So when we put the syringe racks on to the chambers here, they are the ones which are sampling. They are of course in the bottom. So then there will be a small amount. It's about 0.5 milliliters of air in the syringe tube here, which is directly connected to the chamber lid. We deliberately put the syringes as close as we can to the chambers to avoid air or to avoid filling with, for example, molecular water, which makes it more difficult to get accurate values when you analyze. So there is air in these tubes here, about 0.5 milliliters. And normally what we see is that if we deploy at greater depth than about 10 meters, which we do in 99% of the cases, this air after some time will dissolve and will disappear with pressure. We used video to check that, and there is no air then when you sample the water. So yes.
Okay. Any other question? Altre domande? Vabbè, bene, comunque se vi viene in mente una qualche altra domanda me la potete mandare per email, potete contattarci, vi risponderemo anche in un secondo momento. Io vi ringrazio ancora della partecipazione. Thank you, Anders. Spero, mi auguro che quando i tempi saranno migliori riusciremo a svolgere workshop e seminari di persona, e con anche demo e dimostrazioni. Per ora vi saluto, vi auguro una buona giornata e buon lavoro. Thank you again, Anders. Have a nice day and see you soon.
Mille grazie, buona giornata.
Bye, ciao a tutti.